Okay, the next one is types of mutation based on quantity. Quantity we mean how much of the DNA is under the influence of this mutation. First one, according to it, there are two types: gene mutation and we have chromosomal mutation. So we'll start with gene mutation. Since it is a mutation on a gene, okay, just one nucleotide, it's also called a point mutation because it occurs in just one point. Here is the example. TAC is one codon, and this, instead of A, and the next one you have a mutation where the T is translated to, A is translated to T, and subsequently on the other side it has an A. So this is a, mutation, point mutation. And there are plenty of good examples. A classic example would be sickle cell anemia. Sickle cell anemia, which we'll discuss in our next video. Okay, another video. But the point is this: mutation of a nucleotide in the DNA is a gene or a point mutation. That influences just one single base pair. Okay, it is just going to cause harm to one base pair. And this can be caused by two methods. One is substitution and the other is frame shift. Substitution can be of two types again. One is transition, the other is transversion. Okay? Now let me go back to what a DNA strand is. This is the long DNA strand with nucleotides, phosphate, pentose sugar, and a base. So the four bases are classified under two headings: purines and pyrimidines. This you done last year, okay? Purines have double ring, okay? Whereas pyrimidines have only one ring. So if a pyrimidine is replaced by a pyrimidine, that's it. Transition. Or a purine is again replaced by another purine. That's transition again. Transversion, where a purine is replaced by a pyrimidine. Okay? That is what transversion is. Frame shift. Now, this is substitution is just going to affect one pair of your gene. Okay? One pair of nucleotide. But a frame shift is a little different than this. It's caused by insertion and deletion. Now, let's say, for instance, these are the codes. And then there's a deletion of one code. One nucleotide is gone. So instead of these three being read as one codon, now it is these three that is read as one codon. And this will change the type of amino acid that it is going to code for. Okay, so this causes the whole shift in the reading of the codes. That's a deletion. Insertion is the addition of another nucleotide in these non-coded region. So this will again change the code. The codons are changed. The whole frame is shifted. So that's called a frame shift. A frame shift causes a lot more harm than just a substitution. Most of the time, what happens is frame shift causes a production of a protein which is not compatible, okay, or it's not capable of performing the actual designated function. Okay, so this is an incompatible